Coryneform bacteria are a diverse group of organisms, and this video will summarise what they are and how they survive in their environments. They're classified as gram-positive and non-sporulating, and are generally aerobic and non-motile. The group is characterised by its varied cell morphology. Cells are pleomorphic rods, meaning that their shape can vary, being found in V-shapes or club shapes, or changing shape during development. The different coryneforms are grouped into various taxa, which can be shown in a classification tree. All members of the group come from the same order, Actinomycetales, but can be found in different families and genera. Not all of them are listed here. We'll focus on a few key genera. First, Corynebacterium. Many species of Corynebacterium are club-shaped due to the presence of large polyphosphate granules which make the cell appear swollen at one end. These provide energy reserves in times of stress and are thought to play a role in pathogenicity. Various species also go through a process called snapping division, where two daughter cells remain attached by the outer layer of the cell wall and one side ruptures to create a bend. This can occur once to form the characteristic V-shapes, or multiple times to form what is often described as palisades or Chinese letters. Different species may exhibit one or both of these traits, or neither, in which case they're found as a simple rod. Some members of the genus are facultatively anaerobic, but this depends on the habitat each species lives in. Corynebacteria are found in all sorts of environments, including plants, soil, water, and on the skin and mucous membranes of many animals. In all of these, the organisms have to compete with other bacteria. Many of them produce bacteriocins, molecules which kill or inhibit organisms of other species but have little effect on different strains within the species. This helps them survive even in environments populated by other bacteria. The molecules are also heat resistant, allowing them to inhibit sporulating bacteria after a period of higher temperature. When living in animals, some species can present as opportunistic pathogens, and one of these is C. diphtheriae, which is often used as a model for the genus. This organism only causes disease due to a gene acquired from phage. This encodes the diphtheria toxin, which ultimately leads to cell death in the host. Now, moving on from Corynebacterium, let's look at Arthrobacter. Similar to Corynebacterium, these go through snapping division to form V-shaped pairs. However, they also have the interesting characteristic of changing shape during development. Cells in young cultures look like irregular rods, but become coccoid in the stationary phase. When transferred to a new medium, the cells grow to become rods again. This helps them survive throughout the crowded conditions of the stationary phase, as in their coccoid form, the cells are incredibly resistant to starvation and drying out. It's unusual for non-spore-forming organisms to have this quality. A few Arthrobacter species, such as A. globiformis, have been found to be facultatively anaerobic, allowing them to survive in environments where oxygen levels change frequently. Another factor that enables Arthrobacter to live in a wide range of habitats is their nutritional versatility. They're able to use a number of molecules as their sole source of carbon and nitrogen, such as nitroglycerin. The final example we'll talk about is Cellulomonas. These cells can take a wide range of shapes, being seen in coccoid or rod forms, and, like the other genera we mentioned, often undergoing snapping division. Different members of the genus are found in a massive variety of environments, from the ocean to the human spine, but most species live in soil and on decaying plants where there's an abundance of cellulose. Environments like these are where Cellulomonas organisms thrive because of their ability to break down cellulose as a food source. They do this by the use of cellulase enzymes. Many species produce glucanases and xylonases, which hydrolyze cellulose into its constituent parts. Animals which eat a lot of plant matter but can't break it down provide a great niche for them to live in, and so various species can be found in places like the rumen of cattle. Cellulomonas demonstrates a lower ability to utilize cellulose in the presence of other, more easily metabolized food sources, and this suggests that it's a mechanism developed to survive in low-nutrient environments. These three examples should give an idea of how varied the group is. Some species are unusually resilient, growing in ever-changing environments, and many change themselves in order to do so. This includes shape changes, aerobic to anaerobic respiration, fermentative metabolism, and many other mechanisms. Most places on Earth contain Coryneform bacteria, and although species of most genera can be found in soil, many live in more unusual environments, making use of different nutrients as a carbon, nitrogen, or energy source, and, most importantly, they have a variety of different shapes, all of which help them survive in their environment.